This is Shivani Jadhav, student of the Elite Institute of Pharma Skills. My seminar topic is clinical trial phases. Let's see what is clinical trials. Clinical trials is a systematic investigation in human subject of evaluating the safety and efficacy of any new drug. Clinical trials are a set of tests in medical research and drug development that generates safety and efficacy data for health intervention in human beings. There are three important keywords in clinical trials that are experimental units, treatment, and evaluation of the treatment. Clinical trials are conducted only when satisfactory information has been collected on the quality of the non clinical safety. Health Authority Committee approval is granted in the country where approval of the drug is sought, and clinical trials is the best for bringing out new drugs to the market. This is the basic outline of clinical drug development phases. This trials follows a specific study plan called a protocol that is developed by the researcher or a manufacturer. Before a clinical trial begins, researcher review prior information about the drug to develop research question and objective. Then they decide who qualifies to participate in trials, that is the selection of criteria. The drug development process. This is the basic format of drug development process. First step is preclinical testing. Preclinical testing is done on animals. Then IND application, an investigational new drug application, means which a pharmaceutical company obtain permission to start human clinical trials and to ship an experimental drug across the state lines. After the preclinical testing, the clinical testing are performed, in which the phase 1, phase 2 and phase 3 studies are carried out. After the clinical testing, the submission of a new drug application, that is NDA, is formal step asking the FDA to consider a drug for a marketing approval. FDA will approve the application or find it either approvable or not approvable. After the FDA approval, the phase 4 studies are carried out, in which the adverse reaction are reported and the sampling and surveys testing are carried out. Phase 0 studies. Study of the new drug in microdose to derive PK information in humans before undertaking phase 1 studies is called as phase 0. The objective of phase 0 is to obtain the preliminary pharmacokinetic data. Preclinical data, the subaqueous toxicity in one species by two routes of administration. These are very early studies of the pharmacodynamic and pharmacokinetic properties of potential drug in humans. Microdosing approach could accelerate drug development without compromising clinical safety. Phase 1 clinical trials, that is human experimental trials. Phase 1 trials refers to the first introduction of an experimental drug in a human population. The aim of the phase 1 trial is to determine the maximum tolerated dose, that is MTD, of the new treatment. The objective of the phase 1 reaction, that is phase 1 trial, is assessment of the drug safety and safe effective dose of the further phase studies, or to determine what happened to the drug in the human body, or to accelerate or to evaluate how new drug should be administered and the side effects of the drug. The phase 1 general experimental design. The phase 1 studies are carried out in two stages, that is single rising dose and repeat dose administration. In single rising dose, each voluntary given a single dose of drug or a placebo, that is dose escalating study design. Initial dose and route of administration decide from existing preclinical data. 8 to 10 voluntaries admitted after the meticulous screening, and 2 to 4 voluntary receive placebo and 6 to 8 voluntary receive drug under the study. Only after result of first group analyzed, drug administered to the next group. Then repeat dose administration. Repeat dose administration start after the single dose administration result access. The drug or a placebo given repeatedly for one or more weeks. The interval between doses is usually one half life. The study design is typically dose escalating study. The basic condition for phase 1 is preclinical data, IND application, approval by a regulatory authority, protocol approval for ethics committee, informed consent, and adherence to the Declaration of Helsinki or ICH GCP guidelines. Phase 2. Phase 2 trials further assess safety and effectiveness testing on larger groups of patients comparatively. The objective of the phase 2 is to confirm the effectiveness, monitoring the side effects, further evaluate the safety of the drug. It includes two stages, that is phase 2A and phase 2B. Phase 2A is designed to assess dosing requirements and phase 2B is to study the efficacy. This is the general difference between phase 2A and phase 2B. Phase 2A it is early phase and phase 2B it is late phase. In phase 2A 20 to 200 patients are required and in phase 2B 50 to 300 patients are required. In phase 2A single blind compared with a standard drug and in phase 2B double blind compared with a placebo or a standard drug. Phase 3, that is therapeutic confirmatory trial. Phase 3 is usually considered to begin with initiation of studies in which primary objective is to demonstrate or confirm a therapeutic benefit. Generally conducted in large number of patients, about 1000 to 500, and last anywhere from 2 to 10 years. The objective of the phase 3 reaction is to establish the efficacy of drug against the existing therapy, to establish the safety in relative large number of patients, to establish the method usage in clinical practices, and to identify the contradiction and warning for the use of drug. Phase 4. Phase 4 is also known as post marketing surveillance study, that is PMS. It is done after the drug has been marketed. The studies continue to collect the data about effect in various population and side effects from long-term use. The objective of phase 4 is helps to detect rare adverse drug reactions and drug interaction and evaluation of overdoses, quality of life assessment and comparative benefits risk assessment. The summary of the clinical trials is the clinical trials testing potential medical products are commonly classified into four phases. The drug development process will normally proceed through all four phases over many years. If the drug successfully passes through phase 1, 2 and 3, it will be usually be approved by national regulatory authority for use in general population. Phase 4 trials are post marketing or surveillance studies conducted to monitor safety over several years. Thank you.